Hello everyone, this is Dr. Masume Gholami. I'm an associate professor in Department of Physiology at Arak University of Medical Sciences. Welcome to our lesson on cell physiology. I'm excited to have you all here today. In this class, we will explore the fascinating world of cells, their structures and the vital functions they perform in living organisms. This course will consist of 7 sessions and your reference for a study will be the book Guyton and Hall Textbook of Medical Physiology. Let me start the class. In the beginning, you need to know the definition of the physiology. As you know, the physiology is the field of the science that aims to clarify the physical and chemical process underlying the emergence, growth, and advance of life. There are the vast field of the physiology can be divided into for example, plant physiology, mammalian physiology, bacterial physiology, and the human physiology. And the goal of this lesson is to teach human physiology. Now let's begin with the smallest unit of the human body, which is the cells. As you know, the cells is the smallest unit that is capable of performing life function. And we have a different type of the cells in the body, for example, muscle cells, adipose cells, red blood cells, nerve cells, glial cells, and the other type of the cells that located in the body, and these cells come together to form an organ. Cells are organized in the various components, each playing a vital role in their function. We have several key components in the cells, include nucleus, cytoplasm, organelles, and plasma membrane. You know, the nucleus is a control center of the cell and housing genetic material, DNA, and the cytoplasm is gel-like substance that fills the cell and contains organelles. And the organelles are specialized structures within the cell that perform specific functions, and the plasma membrane is the outer boundary of the cell, regulating the movement of the substance in and out of the cell. The composition of the cells include water. You know, the 70 to 85% of the cell is water. That's essential for various biochemical reactions, and the ions are the other composition of the cells that is essential for cellular function. We have a different type of the ions in the cells: sodium, potassium, chloride, calcium, and bicarbonate, and the other type of the ions. The protein is another type of the composition. The protein has a structural and functional role in the cells, and the lipids include phospholipid, cholesterol, and triglyceride. The phospholipid is a major component of the cell membrane. Cholesterol is important for stabilizing the cell membrane, and the triglyceride is a energy storage molecule. Finally, the carbohydrate. The cells, especially we have a carbohydrate in the cell membrane, include glucose and glycogen. Is two important type of the carbohydrate that, for example, the glucose is a primary energy source, and the glycogen is a storage form of the glucose, that especially in glucose and the other type of the carbohydrate is located in the cell. The another type of the organelles in cytoplasm is lysosomes. Contains many different digestive enzymes and provide the intracellular digestive system and allows the cell to digest the main cellular structure, food particles that have been ingested by the cells, and digest unwanted matter such as bacteria. Endoplasmic reticulum is organelles that's located in the cytoplasm, and this organelle is interconnected network of flattened sacs and tube-like structure. We have two types of the endoplasmic reticulum, or ER, rough endoplasmic reticulum or granular endoplasmic reticulum. This type of the endoplasmic reticulum contains ribosome, that which is the site of protein synthesis. And the another type of the ER is a smooth ER or a granular ER. This ER lacks ribosome and functions in lipid manufacture, the production of the steroid hormones, and detoxifications. We have another type of the organelles in the cytoplasm that's called Golgi apparatus. The Golgi apparatus act similar to post office. It modifies, package, and labels item, which it then sends to different parts of the cells or to the extracellular space. For example, proteins synthesized in ER are packaged into the vesicle, which then fuse with the Golgi apparatus. The another structure that's located in the cells is a cytoskeleton. It's a complex network of fiber and filaments that provides the structural support and shape and organize to cells. It plays a crucial role in various cellular functions. The main role of the cytoskeleton are providing a rigid structural support, give the cell its shape and mechanical resistance to deformation, and intracellular support, for example, axonal transport. These transport some vesicle and organelles movement within the cells. And other role of this structure is cell division. for example, the separation of the chromosomes by formation of centriole. Finally, the other role of the cytoskeleton is muscle contraction. You know about the actin or myosin myofilaments that have a function in the muscle contraction. I speak about one type of the intracellular transport that's named axonal transport. Most axons are two-line to allow efficient movement of substance from the soma to axon terminal by simple diffusion. Axonal transport is a cellular process responsible for movement of the different cargoes, for example, mitochondria, lipids, synaptic vesicle, protein, and the other type, and form a neuron's cell body. This movement is bidirectional, for example, from the cell body to the axon terminal or axon terminal to the cell body. When the cargoes movement from the cell body to axon terminal, these movements name a retrograde. And when the cargoes move from the axon terminal to the cell body, this movement is an antegrade. For axon terminal, the microtubules made of tubulin run around along the length of the axon and provide the main tracks for transportation. We have two types of the axonal transportation: antegrade transport and retrograde transport. In the antegrade transport, motoprotein kinesin, which transfers vesicles containing neurotransmitter and enzyme responsible for the synthesis of the neurotransmitter to synaptic terminals. In the retrograde transportation, the motoprotein dynein, which return recycled synaptic vesicle to the soma for lysosomal degradation. The speed of axonal transport is different. Organelles and vesicles are transported by fast axonal transport, and substances that are dissolved in cytoplasm, for example, protein, are moved by slow axonal transport. In a mammalian, fast axonal transport proceeds as rapidly as 400 mm per day, whereas a slow axonal transport occurs at about 1 mm per day. The movement of the slow cargoes is actually rapid, but unlike fast cargoes, they pause frequently, making the overall transit rate much slower. The mechanism is known as the stop-and-go model of slow axonal transport. As you see, this picture shows the molecular machinery for axonal transport. The yellow arrow shows the antegrade and the pink arrow shows the retrograde. As you see, in the antegrade transport, the kinesin has a role, and the dynein has a role in a retrograde transport. And you see the cargo in an antegrade transport, for example, the vesicles, cytoskeletal protein connect to the kinesin, and the move on the microtubules convey from the cell body to the synaptic terminal. But in the retrograde transport, the cargo connect to the dynein, and the dynein move on a microtubule from the axon terminal to the cell body. And you must know the axon terminal requires the metabolic energy ATP and the calcium ions. As a clinical example, the rabies virus reached the central nervous system by retrograde axonal transport, and the tetanus neurotoxin is a toxin block the receptors for inhibitory amino acids, and this process can result in the tetanic convulsions. So these tetanic neurotoxins is internalized at the neuromuscular junction and retrogradely transported toward the soma in the signaling endosome. These articles is about the rabies virus and the axonal transport. If you you can see them. The important component of the cells is a cell membrane. The cell membrane form a lipid bilayer. The lipid bilayer is impermeable to the water-soluble substance such as ions and glucose. And conversely, fast-soluble substances such as the oxygen, carbon dioxide, and alcohol can penetrate this portion of the cell membrane with ease. The cell membrane has three components: membrane lipids that form 42% of membrane, membrane protein that forms 55% of membrane, and membrane carbohydrates that forms 3% of membrane. This percentage is not constant according to cell function and cell type. This percentage can change. For example, in the nervous system, the percentage of lipid, especially in myelinated fiber nerve, is high because of myelin is composed of protein. Opposite in mitochondria that makes ATP in the cell, the percentage of protein that has important role in the ATP synthesis is high. Okay, at the first we want to speak about the membrane lipids. Okay, in the previous slide I told you 42% of cell membrane is lipid. You know 25% of these lipids are phospholipids. 13% is cholesterol, and few percentage of membrane lipid composed by another lipid, for example, lipids bounded to carbohydrates. Therefore, we start with the phospholipids. As you see, phospholipids consist of three parts: head, tail, and neck. The tail of phospholipid has composed by two fatty acid chains, which one of fatty acid is saturated and the another chain is not saturated. Saturated fatty acid have hydrocarbon chain connected by single bonds, and the unsaturated fatty acid have one or more double bonded. The head of the molecule has made by a phospholipid group which can esterify to an organic molecule such as choline, ethanolamine, and inositol, and another type of the like to these molecules. These organic molecules will change in the type of the phospholipid. As you see, for example, this organic molecule is ethanol amine. So this phospholipid has called ethanol amide. If this molecule was choline, the phospholipid has called phosphatidylcholine. And the structure between the tail and head is called glycerol. As you can see in this picture, the cell membrane is a bilayer, and the basic structure of the lipid bilayer is composed of three main types of lipids: phospholipids, sphingolipids, and cholesterol. The head of phospholipid is composed of phosphate group, which is soluble in water. This makes it hydrophilic, while the tail fatty acid chain of the molecule is hydrophobic, because the hydrophobic portions of the phospholipid molecule are repelled by water but are mutually attached to one another. They have a natural tendency to attach to each other in the middle of the membrane. Consequently, the hydrophilic phosphate portions constitute the two surfaces of the complete cell membrane, which are in contact with intracellular water on the inside of the membrane and extracellular water on the outside of surface. As a result, the lipid bilayer in the middle of the membrane is impermeable to unusual water-soluble substances such as iron, glucose, and urea. Conversely, fat-soluble substances such as oxygen, carbon dioxide, and alcohol can pass through this portion. The cholesterol molecule in the membrane are also lipids, as their steroid nuclei are high fatty soluble. These molecules are dissolved in the bilayer of the membrane and mainly help determine the degree of permeability of the bilayer to water soluble constituents of body fluids. As I mentioned that addition of lipid, the cell membrane composed by protein. The cell membrane protein, according by a structure, divides to two types: peripheral protein and integral protein. Integral protein that protrude all the way through the membrane, and peripheral protein that are attached only to on surface of the membrane and do not penetrate all the way through. As you see, the left side of the picture shows the peripheral proteins. Peripheral proteins are not covalently or loosely bonded to integral protein. These proteins do not reside within the thickness of the cell membrane, and they can be separated from the membrane by altering the concentration of salt, and we don't need destroy the structure of the cell membrane for separation of this type of the protein from the cell membrane. In contrast, integral protein are embedded within the lipid bilayer, residing either wholly or partially within the membrane's thickness, and to separate these protein, detergents must be used. and which the detergent disrupt the membrane structure, and for separation of these type of the protein from the cell membrane, we need disrupt the membrane structure. Protein have important function in the cell membrane. For example, integral membrane protein can serve as a receptors, adhesion molecules, transmembrane movement of water-soluble substance, enzymes, and intracellular signaling. And the peripheral membrane protein participate in the intracellular signalings and form a submembranous cytoskeleton. The other main component of membrane is carbohydrate, and I told you carbohydrate constitute 3% of membrane mass. All of carbohydrate in the cell membrane attached to lipid or protein and form the glycolipid and glycoprotein respectively. And all of the carbohydrates in the cell membrane are located in the outward from the cell surface. The all of carbohydrate in outside surface of cell membrane is called glycocalyx. The carbohydrates can serve as receptors and membrane protection against chemical and mechanical factors, can cause adhesion of cells to each others, and participation in immune system function. Finally, as a summary, this picture shows the three important components of the cell membrane. I told you the cell membrane form a lipid bilayer. The main lipid of the cell membrane is phospholipid, and we have two types of the protein in the cell membrane: integral protein and the peripheral protein. Another component of the cell membrane is carbohydrate. This carbohydrate connect to the protein or lipid and produce glycoprotein and glycolipids. Okay, thank you for your attention. I think that's enough for today. I hope to see you in Iran. Goodbye.